So yeah, the title of my talk, as David was mentioning, is all about microbial communities in the ocean and using high-throughput sequencing. I'm going to give you just a brief introduction or of the structure of this talk. So I will give you a general introduction. We'll talk a little bit about high-throughput sequencing, then about computing power, and in the end, about aquatic microbial communities. As you probably know, in Google, they have been sort of scanning a lot of books from all around the world. So it's a nice exercise to look about the interest people had in different subjects during the last year. So this is just like a general background information. So if you look, for instance, in bioinformatics, there's a small increase recently. DNA sequences had a big increase since the 70s. And then in informatics, has a very large increase during the last, let's say, 20 years. In marine ecology, for instance, has been here, like, it has an interest, but it hasn't been growing so much. So this is a general background just to show the interest there has been in the subject. Now I'm going to mention a few things on this field, like a few landmarks. As you know, in the 50s, we have the discovery of the double helix. Then we have the first DNA genome in the 70s. Then we have also the publication about the Sanger method, the first commercial sequencing machine in the 80s, and so on. Then we have the pyrosequencing published in 96. In 2009, we have the first human genome for $50,000. And recently, we have the first human genome for about $1,000. So if you see in about 60 years, there has been a huge increase in, or a huge advance in this field. This is a very brief chart on sequencing approaches. So I won't go into detail in this. Just to tell you that until 5 years ago, most sequencing was done here as the Sanger method, the typical one. Now we have the high-throughput techniques, which are the most popular are these ones. You have other ones as well. But there are different methods, but these are just to give you an idea of the general approaches. Another thing which has been quite striking in this field is the decrease on cost. So in about 10 years, as you see, you went from cost per genome of like 100 million to about few $1,000s. So things are decreasing pretty fast. And the same thing you see it, well, for the prices on raw megabase sequencing. So now I'm going to talk a little bit of high-throughput platforms. And as I mentioned before, this is the first generation, what we call the Sanger sequencing, which produce about like 10 to the minus 6 of gigabases of output. It produces longer sequences, like 100 base pairs. And if we put like, if we cut this here, now we start with the next generation sequencing or current generation sequencing because they have been here for a while. So the first one, these are the popular platforms. So a quite popular one is 454 from Roche, which produce about 0.4 gigabases of output and sequences from 500 to 700 base pairs. And now we have Illumina as well, which, look at this, it's 500 gigabases of output. So there's a gap here. Among next generation sequencers, there's a large difference as well. So this produces a huge amount of data. On the other hand, it produces shorter reads. And these are the so-called third generation sequencers. Like this is a PacBio machine, which is a single molecular sequencer. And well, the throughput is smaller, of course. It produces like about 3 gigabases, but on the other hand, it produces longer reads. And these are other platforms. Probably you've heard about ABI Solid, Ion Torrent, which is like a coming technology. I think we've been now during the coffee talking about this one, the Nanopore, which, well, it's a single molecule sequencer, which is not yet commercially available, but it's promising quite a lot. And if we look at cost, this is the cost you have for Sanger sequencing. And then this is the cost per megabase in Europe. If we move to the 454, the decrease is pretty high. And then look at this. These are all other next generation platforms. It's really low. And here we see a map. Let's say if we map all the machines around the world, well, this is the distribution of machines. As you see, well, machines are clustered in the usual places, like North America, Europe, and Japan. And this is a map for Spain. There are like 22 in Barcelona, 6 in Valencia, 3 in Madrid, and apparently there's one here. I saw the name, but I'm not sure where it is, this place. I think it's a hospital or something. This is numbers in the world of sequencers. We go from, I mean, the two most popular platforms, Illumina, probably sort of monopolized, no, not monopolized, but they are the most popular in the market. Then we have the ABI Solid, and then Roche, and then some other ones. And during the last years, there has been a lot of public interest or in the press about next generation sequencing. If we look at the amount of data produced now in the world, these numbers are quite astonishing. There are about, more or less, about 1,700 functional high-throughput sequencer machines of different brands. And this should produce about 13 petabases of data, which is about 10 to the 15. So it's a lot. It says about a million human genomes per year. The thing is, now the question that comes to probably most of you is how do we handle such amount of data? And this brings us to the next topic, which is computer power. So now we switch a little bit of topic. You probably heard about this, about Moore's law. This is the guy from Intel that, I think in the 60s or 70s, produced this law saying that microchip power doubles more or less every 2 years. And this is a little less known Cridor law, which is storage capacity doubles more or less annually. But look at this. I mean, the improvement in sequencing output is improving much faster. So if we have the Moore law here of decrease, let's say, in cost per computation or something like that, if we compare to the decrease in cost per sequencing megabase, there's a large difference here. So the worldwide capacity is this is more or less estimated, is growing at about 4 or 5 times per year. This is pretty high. So here I'm showing just a few other relevant facts, just to keep this in mind. So the base per base costs are dropping about half every 6 months. Only one of these sequencing platform, the high-throughput 2000 from Illumina, produce about 20 terabytes of data per month. This is like 20 hard drives that you use for moving data around. So 20 of these are produced every month. A large research center, I took this data from this Cold Spring Harbor lab. They have about 1-2 petabytes of storage for the whole institute, which is about 2,000 terabytes of data. And the problem is that storing all these outputs from high-throughput sequencer is becoming an issue. You need to know what to do with the data. You cannot store that forever. Would you delete that? Will you keep that? What to do? I mean, it costs money to have all this data you occupy in hard drives. Another thing which is happening is that some public databases are becoming saturated, like the typical NCBI, where probably some of you submit some sequences every now and again. For a while, they were like sort of wondering if they would continue accepting high-throughput sequencing data because it costs a lot of money. Yeah, it's expensive. So now we see a bottleneck, which how I see it is that the data processing part and the computation part are some current bottlenecks. I'm going to tell you a little bit about the computer power you need now. This is a very rough estimate about what you need now to process data. So as you probably know, well, with a common laptop, you could process your first generation sequencing data, your Sanger data, was no problem. So if you move to let's say second generation, like 454, you probably want to have at least a small server. You know, a few cores, like from at least, I don't know, 16, a few gigabytes of memory, and a few terabytes of hard drive. Now, if you move to Illumina, you most probably need a cluster. You cannot really do much in a server like this. This is the one server I have, and when I tried to process Illumina data, it was a mess. It was painful. So you probably want to move to a cluster. So I would say for Illumina, you most likely need to move into a large machine. And for some third generation sequencers, well, you might do it in a small server or directly in your cluster. These are another option for if you have very intense computations, let's say a very specific task that one script or one pipeline has to do, you can move to this GPU processing, which is their graphic processing units, where you have a lot of small cores or processors, let's say, and you can run serially, parallelly, all your computations. Another option, which I think it's pretty promising, it's cloud computing. In here you can buy the amount of computer power you need. So you buy it and then you forget about it. It's very scalable. So if you need 100 processors, or if you need 10,000, as long as you can pay it, you can use it. And you don't have maintenance cost. And one good example is the Amazon cloud. And then there are some platforms which are quite user-friendly, like DNA Nexus. I think we've been talking yesterday about this one, or Neat Refill, which are designed for high-throughput sequencing analysis, and the analyses are sort of channeled into the cloud. There are probably more around. These are the ones I saw. I haven't been doing much in the cloud, but it looks really good. Probably in the next year we're going to do some more things there. And this is just to show you one machine. I feel very happy to be able to use this. And this is actually how I met with David, because he was, reviewing applications, and then, we became connected by that. And this is a large computer, like a supercomputer with I think it's about 10,000 cores. And yeah, it works very nicely, and it's located, as you see, in a church, which in a way is telling that, Barcelona, science and religion, it was pretty nicely. I don't know if you can go and pray into that church, but maybe. So just to summarize a bit, I would tell that high-throughput sequencing has a great impact or a great potential in diverse fields. Let's say medicine, agriculture, like improvement of livestock or things like that, biotechnology, doing gene discovery. There's people talking about, in a few years giving your genome as a gift or stuff like that. I'm not sure if that's going to be happening or not, but it's around. These people is talking about this. Global chain biology, there are some interesting papers coming out in that, looking at how, let's say, global warming might affect microbes in global change. And also in environmental microbiology, which is more or less my field, aquatic and terrestrial. And, this moves us to the next topic, which is aquatic microbes. So maybe some of you wonder, why to study aquatic microbes, why they are important? And I guess here in vivo, since you are kind of quite connected to the sea, it's a relevant question in a way. If you look sometimes from space to the Earth, you see things like that. And then you wonder, what's that? Well, that's a microbe. And then you think, if you can't see this from space, then microbes should be important, at least for something. And they are. And they are important for global ecosystem functioning, for biogeochemical cycling. Biogeochemical cycling is the movement of elements, of chemical elements between the living and non-living part of the world, and as well for evolution. this is a very nice slide where sort of try to integrate these concepts. I'm not going to go like in detail in all that, but let's say that microbes, they have very important roles in the base of the food chain. So they connect, well, they are probably primary producers, and they are the base of all these food chains. They are also very important for several different metabolic processes occurring in the community. And the link of microbes and all these metabolites is their DNA that they carry. So if you look at the specific spot in the sea, I mean, things like this are happening. You have a community metabolism, you have a food chain, you have DNA working in this microbial community. But then if you extrapolate this to the whole ocean, you start to have processes which have impact at the global level. So let's say you have the ocean microbiome, where you have the DOC, this dissolved organic carbon, being taken by microbes. Then you have a cycling of nutrients. Then there's absorption of the CO2 from the atmosphere. There's emission of gases. So at the global level, the ocean microbes, they have an impact. This is another nice slide I took from this very good paper, I think, where you have, let's say, geochemical processes occurring here and chemical elements being produced here, which move into this compartment, which is the living part of the world. This is actually called a model of the biosphere, like inorganic elements taken by or cycling by the biosphere, being released. So there are a lot of processes which are mediated by microbes. And also, microbes are really important for understanding evolution. If you look, this is a very simple chart. Most of the evolutionary history of life occur at micro scales. So there's a lot of information there for us to extract and for understanding evolution. And there's a great potential in microbes, since so much evolution occurs in them, to find new genes or novel genes. So the previous was like a very general overview. Now let's focus more a little bit. What can high-throughput sequencing tell us on microbes? So here are some things I'm particularly interested, but this is not all you can do. There are probably many more things you can do. This is just a few things. So high-throughput sequencing, I think, is very useful to use for understanding the extent of diversity, taxonomic and metabolic, to understand spatiotemporal distributions, evolutionary and ecological patterns, and as well as interactions and metabolisms. One question that has been around for quite a while is related to microbial diversity and distributional patterns. One thing is how many microbes there are, which types you have, and also how they are distributed in space and time. And this is quite interesting, because if we go back about 200 years, people were going into ships, like Darwin going into a ship and went around the world trying to find what plants and animals he could find in different continents. And this is just a few years ago on, let's say, the first large expedition to search, in this case, for microbes. So there's a parallelism here. It's 200 years apart. We are in the microbial world, let's say, in a similar stage as what Darwin was 200 years ago. So now I'm going to tell you about one of these global expeditions we are involved. It's called Tara Ocean. So we are doing work in Barcelona, and this is a consortium with a lot of French people. It's actually the headquarters are in France, and it's organized from there. And it's basically a small ship that is going around the world and sampling the ocean. So how is Tara sampling the ocean? Well, it's dividing the life in the water in different size fractions. I think probably, well, most of you are kind of familiar with this, but what you do is you say, well, you have all this is life in the water, and you allocate it in different size fractions. So you have the virus fraction, the bacteria fraction, the microplankton, and so on. And for that, you use different devices to filter or to get the different fractions. Well, typical devices are like this. You use nets where you have different pore size, so you capture different parts of the microbial community or bigger fractions, and you use well, these bottles that you can collect water from different depths and so on. What Tara is doing afterwards is doing a lot of sequencing on those fractions with doing DNA, cDNA. Well, it's a lot of information being extracted there. And it's also a lot of high-throughput imaging, but I'm not going to go into this. Now, you have all this DNA information, but you want also information from the environment. That's very important. You need to link afterwards environmental data with DNA or genomic data. So there are other things which are other parameters, environmental parameters, which are measured in this project. And if you wonder how it's to work in a ship, well, this is people working in a ship, Isabel and Hugo, who are doing serial filtration of water, and you collect all the microbes in large filters, which are in this called ovnis, and then you freeze those, and then you keep your DNA there. So far, Tara is like coming to an end as a project, and there are quite many station samples around the world. What I want to tell you now is that, well, you have imagine that you went into a cruise like this, and then you have all your DNA, and then you wonder, well, how do I proceed now? So I'm talking a little bit about method. This is very, very simple. These are two approaches that you could use. Let's say you have your community DNA, you're in the lab, and then you wonder, well, what to do? You can go into a kind of, let's say, traditional way, and try to target a specific gene that you're interested in, amplify it, and do high-throughput sequencing. You end up with an amplicon library, and your amplicon library will have either a functional gene or probably a taxonomic marker. A different thing that you can do is like you can do shotgun sequencing, that is, sequence everything that you have in your sample. You produce a metagenome, and that metagenome will have metagenomic reads, well, we have general reads, and we have also taxonomic markers. One thing which is possible now, or more feasible now, is to generate large metagenomes, because you have a very high throughput now. And that metagenome will have both. We have all reads or all sequences from the community belonging to many different fragments, and you have also a good amount of taxonomic marker. So in this project, what I'm going to mention now, what we did is basically this. We took these two pathways and we asked, well, if we look at the 16S, how we can compare, or how comparable are these two ways of analyzing data? So we took a 16S from amplified libraries, and we also took 16S from metagenomes. These are the guys from Barcelona who I'm working with. Well, he's from the MBL in Germany, and also quite a few people involved from the Tara Oceans. So if you do 454 target sequencing, you get usually reads about 200, 400 base pairs. You will have a PCR bias because you use primers, you use PCR, so things deviate a bit. You generate probably a few chimeras. So if you have a 16S fragment, what you end up is with something like this, several fragments covering one specific area of a 16S. 16S is usually divided in several areas. If you do this second approach, you will have a metagenome. You extract your 16S genes from the metagenome. So you will have, if you do Illumina, you will have shorter reads, about 100, 200 base pairs. The good thing is that you won't have PCR bias. You will have pair-end data, so it's sequencing from both ends, and then you can overlap the reads. But your reads will sort of be distributed in several areas of the 16S. You won't have this scenario. So for this first study, what we looked was at three pilot stations. We have different size fractions, like for bacteria and for larger microplankton. We have metagenomes for all fractions with Illumina and 454. And we also had 454 tag sequencing. And this was for exactly the same samples. So this is an overview of the data set we produced for these three stations. If you look, I mean, Illumina produces a lot of data. It's about 179 gigabases, which is about 1,700 million of reads. From all those reads in the metagenome, about 600,000 were 16S. So we are only concentrating now in bacteria. There were also protists, but we haven't looked at them yet. So if you look at the recovery percentage, it's about 0.03%. This is the percentage of 16S that you can expect from your metagenome. If you do a 454 shotgun, this is about, let's say, 8 runs. So 8 runs of 454 will give you about, 8 million sequences and 3,000 16S. As you see here, there's a difference. I mean, you can probably do a nice community analysis with 600,000 reads, but it's going to be much more difficult with 3,000. And this is about 8 reads. If you calculate about, I don't know, might be about 8,000, 7,000 euro per read. It's quite a lot of money. So for metagenomics, this is, well, it's more difficult approach. I mean, Illumina seems better for all metagenomic approaches. But if you do targeted 454 tag sequencing, well, you can get, well, higher numbers, but you only will get your gene of interest. So when we got all this data, we started to do comparative analysis. And this is a classification now with RDP, which is like it's a good classification algorithm. And here we see the mean and the standard deviation of the classification of all reads across different taxonomic ranks. So as we see, of course, when we want to go lower into the classification quality, which is here, it drops. Still, we see here it's Illumina, here it's 454 tags. Well, I mean, of course, 454 tags do better because they are longer, they provide more information for classification, but there's not a huge gap. So we think that still shorter Illumina reads can be classified and used for extracting all this taxonomic information from communities. Another thing which is interesting is that, well, if you do a PCR, of course you will be using primers, and those primers won't pick up everything in your sample. Probably will pick up a lot, even if you use universal, but not everything. So we expected that from metagenomes we would recover a larger diversity. And this is more or less what we are seeing. For instance, if you look at the order level, just to say something, we see that there are 79 or 70 orders which are picked up by tags, which come from a PCR, but the reads that come from the metagenomes in the 16S, they pick up an extra 33 orders. And this is happening for all ranks except genus, where apparently tags recover a few things which were not present in Illumina. But in general, it seems that this approach of extracting diversity data from metagenome, well, you can recover more from your community. This is just to show a general graph. There's no very specific information here it looks, what I want to say is that it might look more complicated than it is. It's a typical rank abundance. So for each, so what we've done is to cluster all these reads into what we call OTUs, which this is a very rough, let's say, you can use it as a species estimator, but there's a long way from an OTU to a species. But let's say it's kind of a species. So we cluster, things are 99%, and then what we did is to look at the abundance of those OTUs. So here you have the rank, which is, well, if you're first, second, or 10th, according to your abundance. And here is abundance. So most abundant things appear here, and then things drop. And this is observed in most microbial communities. You have a few dominant taxa, which sort of dominate the community, and then many rare ones. And we'll come back to this concept later in the talk. So what we saw is that if we focus only in one area of the 16S, which is the B1, B3 region, either when it's normalized or not, we get more or less similar curves. In this case, things are different because, well, we didn't normalize and we have much more Illumina reads than tags. Another thing that we were seeing is that, well, this is a rarefaction curve, probably you've seen it. Here you have, again, it's not observed species, it's OTUs, but let's say considered species. And this is the amount of sequences that you sample. Let's say that you have a fixed amount of sequences per sample, and then you resample that in order to get an estimate of increase in the number of species you see. It's a typical technique to see if you are saturating your diversity or not. If we focus into the B1, B3 regions, and we use, this is something we did just to make the data very comparable. We cut the 454 reads, we cut it, and we let them have the same size as Illumina. And we see some difference there, but we think these are biases introduced by the short sequences. The good part, despite there is some kind of difference, things are not completely different. We are not getting like very different diversity estimates when we use Illumina 454. So the techniques are comparable in a way. And here we see again this rank abundance. So again, the rank here and the abundance here, and all the OTUs were plotted, well, according to the rank. In green, you have Illumina. And what we've done is to say, well, assuming we plot the rank abundance of Illumina, let's plot then the corresponding OTU and the abundance you get with 454. So let's say if you take this OTU, you get this abundance when you use Illumina, and then the same OTU gets a lower abundance when you look at it with 454. And this seems to happen with a few of them. And then when you move to the more rare OTUs, you see that in some cases, several of them with 454 tags, they get kind of higher abundances. So it looks like there's something there. So we look a bit more in detail, and we separate the information in different stations. So station 7, 23, 30, you have surface, you have DCM, probably you know what's DCM, it's the deep chlorophyll maximum. It's a maximum of chlorophyll that usually occurs in deeper waters. And what we have here is the ratio between between tags to Illumina. So rank here, a ratio here, a ratio of abundances, and we see always this pattern, which means that the abundant OTUs, which are near here, seems to be more represented in Illumina, and the rare one seems to be more represented among the tags. And we think that this is kind of related to the PCR processes. But still, this is a work which is ongoing, so it might change a little bit our conclusions. We've been discussing quite a lot, but we are not 100% sure about this. Another thing we've done is to, these OTUs, so to generate the OTUs, what we've done is a process of mapping the reads to a OTU reference dataset. This OTU reference dataset are known and well-curated long Sanger sequences with which you can generate a large phylogenetic tree. So in this tree, we map the abundances. It's not super clear, but well, you can imagine that there are abundances all around. You have Illumina in green, and in salmon you have 454 tags. So these abundances are mapped on the tree. But what is more interesting is that you see that there are some areas, like here or there, which are like small groups, or you can say even lineages of bacteria, which are present or which are recovered in Illumina, and they are not really present with 454. So you can think on a sort of bias by the primer. So just to summarize a bit this part, if you use tags, we are not trying to be like favor one technique over the other. We want to say that Illumina is a good alternative. Anyhow, if we look at the 454 tags, it's a quite popular technique, which is a positive thing. It produces longer reads. A negative thing, it's like it has a PCR bias, and you can amplify one or a few markers. While with using Illumina on metagenomes is a good alternative for community profiling. You don't have the bias, the PCR bias. Once you use the 16S, you have all the other information that you can use for many other things, for like metabolic profiling or whatever. But the negative thing is that it produces shorter reads. And as David mentioned before, well, this is just one slide to show you that there was another expedition called Malespina, and it's done well. It's been around the world as well, but this one has been focusing in the deeper ocean, and yeah, probably in the next few years there are going to be interesting data from that. This is another picture which is, I think, pretty nice, and we've been talking about bacteria, but there are other players in the waters which are really important, which are protists, microbial eukaryotes. They can also have you can see them from the air or from the space. So they are important players in marine systems. They usually have really complex or more complex genomes with many genes. In general, the general diversity is kind of poorly known. There's been a lot of work done in morphology, which is very good, but it's probably much more which is not known out there. And in many cases, they are uncultured. So we are involved in this project, which aims at looking at protist diversity, and it's aiming to look at protist diversity in Europe coastal waters. So we have. Well, there are 9 coastal stations in Europe, but I'm going to talk about only 6 of them. Samples were taken from subsurface, DCM, sediments. We look at DNA, cDNA, mostly focusing on the 18S. We've done mostly 454 and some Illumina as well. And this work is done with Ramon Massana and other people from France and the Biomars Consortium. Within protists, we focus in an important lineage called Stramenopods. So, for instance, diatoms belong to Stramenopods, so it might sound familiar to you. It's a major eukaryotic group. There are about 100,000 known species, multicellular, unicellular. This is a general tree, very summarized, let's say. All the photosynthetic ones are here, and this is a huge part. But we are not focusing in the most popular Stramenopods, but we focus in some very poorly known groups, which were mostly discovered by Ramon Massana in Barcelona, and he called them MAST. And yeah, that's it. They are MAST 1, MAST 2, that's all we know. One are here, the other ones are there, and they look like this, a ball with the flagella. The thing is that these flagellates, which we can be considered a microbial dark matter, well, they are small groups which, or sometimes they are larger groups, but they are not related to any of the major groups. They are quite basal in the phylogeny, so we might speculate about the early origin in the groups. They have a very wide distribution in the oceans, and they can be ecologically very important. Despite they are very little known, these flagellates eat a lot of bacteria, and they also transport organic carbon to the upper trophic levels when they are eaten by other organisms. So they can be up to 35% of the heterotrophic nanoflagellate community. And well, these are like, so far there are 11 groups defined, and there might be a few other ones. But if we go back a little bit, the biomarks dataset using 454 titanium and the B4 region of the 18S, what we had was about 1.3 million reads, and then from stramenopides we had about 200,000, and then from this mass group we had about 27,000. So you see the numbers of, at least for this 454 dataset, when you compare to Illumina, it gets smaller. One question we had at the beginning was how much novel diversity you find with high-throughput sequencing. One speculation was, as you start using high-throughput sequencing, there's going to be like a universe of diversity coming out. But still, well, I will show you now that we didn't find that. But what we did to explore this question was, well, we said, let's take all the stramenopides we find at NCBI, then take only unique sequences which have this length, and generate trees which have this amount of sequences. And after we have these large trees, construct with longer reads and that we can trust, we can map all our shorter reads, which are about 66,000, to this tree. Well, the tree comparison will come a bit later, but now I will show you something else. What we did here was we did a clustering of the sequences at different threshold levels. So we have 100%, 99%, 98%. We look at, well, clustering threshold, and then we saw the number, we look at the number of OTUs, or so-called species, that we were getting. If we look at the complete stramenopile unique sequences using 454, we saw that the diversity we were recovering was like this line. But of course, I mean, if you have more sequences, you will get more OTUs. So then we subsample this 454 dataset to the same size as the Sanger dataset we had. And if Sanger data and 454 data have the same size, at least for this dataset, we were getting similar diversity estimates. So 454 and Sanger recover similar OTUs at the same sampling depth. So this would be the area of extra diversity that you would say that you might recover using 454. But coming back to the previous question, when I was saying, well, how much new diversity we are getting, what we did was we blast all the reads. Blast is like we compare all the reads to the previous information. And then we got the best blast hit, which is like the most similar thing we could find. And we plot the best percentage of similarity. We did like a density distribution. And what we saw is that most things we were recovering are similar to things which were already known. So we were not discovering like a complete world of new diversity. Most probably what we knew from Sanger sequencing was quite accurate in a way. Then we look at other patterns. We generated some phylogenetic trees with the reads. I'm not sure David will like this because they were like shorter reads, and it's like a quick tree for getting, let's say, indicators or things that can tell you things on groups, but of course should improve with longer sequences, but at least it can give you a general overview. So we see that different groups, they have different amounts of phylogenetic diversity, of different amounts of evolutionary diversification occurring in the groups. And another thing we looked at there was whether we could saturate the diversity of these groups. So one question that is going on microbes is, well, that as you sample more and more, diversity keeps increasing, and where is the limit? Where do you say, okay, I'm saturating diversity now? So at least with these flagellates, if we cluster, so here is the number of OTUs, here is, well, the sequencing depth, the number of reads. If we look at the 98% of clustering, let's say we don't look at this because there might be some errors in the sequencing there, but if we look at the 98%, we see that most groups kind of reach saturation. So it's not that diversity is increasing indefinitely. And in this case, now I come back to what I was mentioning in trees at the beginning, is what, I mean, this is the results of the mapping of the Sanger reads into a core tree. So we produce a long core tree with longer sequences, or a large tree with longer sequences, and then we map all these extramonopile reads onto that tree. Then we took out each of these mass groups. This is a tree which is built based on longer sequences, and then we map all the reads to those trees, which is it's a program called pplacer. So it's a good approach or quick approach to see where in your well-defined phylogeny your reads are falling. So in some branches of the tree, you have a lot of reads that we're mapping, and in some other areas of the trees, you have none. So let's say in some cases, well, some groups are bigger, some groups are smaller, but let's say that some branches of the tree attracted a lot of reads, which, well, in a way it says that if you increase the diversity of Sanger, probably you won't get like these large accumulations. There might be some issues, or we might need to tune better the assignment program as well. So far we could say that high-throughput sequences support groups which were already defined, and we could say that the short reads produced with high-throughput sequences could populate these larger phylogenies built with longer sequences. So it populates those trees. And so far what we see is that the Sanger sequences that are present in the databases, they have some information that is not present in our 454 datasets, and the other way around. We probably recover some 454 data, it's not present in the large dataset. So it's, there's a diversity gap there that we need to fill. Now I'm going to show a few things on diversity patterns, now based, talking about a bit of biogeography, say fraction, depth, based on all this 454 data. So this is a very general graph. It's like we have the percentage of mass reads per sample. So you say you have a big sample, you have a lot of reads, which percentage you have of your organism of interest. And then you plot that for the different stations we have, like Roscoff, Oslo, Naples, Gijón. And we see that there's a variation, but this is putting everything together. So now we sort of separate each group. So we have the different mass groups. And if we plot them, we also see that there's a variation in different sizes. So it's not that you can find more or less the same thing everywhere, but there seem to be patterns. We need more, probably we need more data to understand specifically what patterns are we seeing, if this is temporal, if this is spatial, but at least we see that there's not a uniform kind of distribution of them. Another thing what we saw is that at least if we look at the mass communities in different areas, in general, they tend to be different. Only in between Barcelona or Blanes and Gijón, there was no significant difference among them. But in general, communities of mass seem to be different, and they contain apparently different amounts of phylogenetic diversity. We look also at DNA and RNA, so this could be kind of a proxy to see how active are some things or some flagellates in different environments. But in general, we found a few significant differences between RNA and DNA. So we have here different size fractions, like 0.83 where you capture flagellates, the 320 where you capture them but it's bigger, and we have also sediments and the sediments here. So in this case, for instance, the RNA in the sediments was higher than DNA, so we could say, well, maybe this group MAS8, maybe this group is active in sediments, or it's not just DNA which is sinking there, but it's active. It's different to this pattern where you see that there's a little more DNA than RNA, so we're not completely sure what's going on with these groups in sediments. Another thing we saw is that in some cases, the communities that you get in different size fractions seem to be different from a phylogenetic perspective. So there might be slightly different lineages with different sizes. So a few conclusions. High-throughput sequence didn't find like a large amount of unknown diversity. We found a new diversity, but it's not that there's a huge amount, apparently, at least for this group, that is not known out there. We added quite a lot of intragroup diversity, so we sort of populate the tree. In a way, imagine that you have the trunk of the tree, and with the small sequences, you add a few leaves. Diversity seemed very partitioned in spatial scales, and different size fractions appear to harbor different lineages. And mass also were moderately active in the water column and some in sediments. So, in a way, these are very general conclusions, but remember that these groups are very little known. You have a few pictures on the microscope, a few zones with fish, and we don't know much about them. So we think that with this method, we can start to extract information about very little known groups and start to know them. So now I'm going to talk about the last story. I just realized I've been talking like for an hour now, so I'm going to try to go faster through it. So this is more microbial community ecology. Now I'm going to focus in bacteria again. So one general question we also have is how microbial communities assemble in nature. Why you have the communities you have in the different spots. And this is something we were talking in the coffee as well, that different body parts apparently have different communities of bacteria. You have like a body bacteria biogeography in a way. What you have in your mouth is different what you have in your hands or your feet. If we look at the general ecological theory, spatio-temporal patterns in distribution can be caused by different processes. You have the typical environmental filtering, which in microbes could be associated to this this tenet that was said long ago, everything is everywhere but the environment selects. This could be associated a bit to environmental filtering. Then we have more like neutral dynamics, which is not very dependent on really the environment, but it's more related to stochastic processes related to speciation, extinction, migration. You could have other things like resource specialization, when an organism specializes in something in one area of the world and similar species specialize in another different thing. And then you have a role also, or maybe you have a role played by history, like past dispersal limitations, evolution that could generate patterns in spatio-temporal distribution in species. So we've been doing a work in lakes, because lakes are pretty good for community ecology questions, because basically a big difference with the ocean is that you can define the limits of a community. A lake is a lake. In the ocean, well, it's the ocean. Where's the limit? I'm doing this work with these people. They're in Uppsala and other guys also there. We did this study in an area we think it's really interesting. It's in Antarctica. It's called Vestfold Hills. It's an area which has about 400 square meters, hundreds of lakes in this area. The majority of lakes are marine derived. It means that they were in a way connected to the sea at some point of their history. they either emerged from the sea or they were flooded by the sea. Most lakes are ice covered most of the year, so 1 month per year more or less they are ice free. The area has about 10,000 years, and well, we're not completely sure about this, but we could hypothesize that the migration from other continents might be low, but not necessarily. It might happen that for some groups this is not true. If we look this at from a little evolutionary perspective, this is the area from the air. You see, this is the Antarctic plateau, this big ice sheet that covers the whole Antarctica and continues somewhere here. But this is the ice-free area you have. This is the sea, and these are lakes. Well, this is not the lake, this is the fjord. These are the lakes. And the lakes are like a spot of waters which were isolated. Like, you know, there are no inlets, no outlets, they are ice covered. Well, this is summer, so now it's ice-free, but most of the year they're ice covered. And they are marine derived because they, at some point, they were connected to the sea. So if you look at this from a kind of evolutionary or maybe population genetics perspective, you had the beginning, 6,000 years ago or something like that. You have a homogeneous environment, and then marine microbial communities which were isolated in the lakes, they started to experience different conditions. And why they started to experience that? Because each lake started to develop different salinities just for physical-chemical processes. So this lake, and this lake, this can be almost freshwater, and this can be hypersaline. And the whole variation is occurring here. So imagine, same community exposed to very different environmental conditions. These are some pictures just to relax a bit of so much information. So these are the lakes when you work them in summer. Some lakes, as you see, are really near to the sea. You usually work there by helicopters because there are no roads or anything. And in some lakes, it's quite interesting because you filter, for instance, you filter, I think this was like 50 milliliters of water, and you get this, a 0.2 filter which is clogged by green stuff. And so in some of these lakes you have like soups of microbes. The Antarctic data set in total had, well, we have 17 lakes. These lakes go from freshwater to hypersaline. We also included a data set from Antarctic freshwater lakes just to make a contrast. And in total, we have about 600,000 clean 454 titanium reads from also the 16S and the B3, B4 region. The questions we had in very general terms was, what was the response of marine communities to environmental change? Once they were, I mean, they originally were marine, then they were exposed to very different conditions, so what happened? Then, what happened with rare and abundant taxa? I will come back to this thing, because I have to tell you about why we think it's interesting to look at rare and abundant. We also want to see what processes shape communities. And this is a very interesting question. I come more from like a evolutionary background, so I tend to think a lot on the past. But my colleagues in Sweden, which are microbial community ecologists, they said, well, I mean, the whole community might have lost all the connection with the past. What you see now, it's the outcome of the last 5 years. All the past might be gone. And this made me think quite a lot, and this is something kind of new way of thinking. It's like, if you go to South America now, you might see like European people. You don't see the people that were there before. So another question we had was, can we detect habitat specialists and generalists? This is, are organisms more associated to specific habitats or not? And the last question, well, had to do with novel diversity in Antarctica. So what we did here is to fit our data to this neutral model. The neutral model would say, well, the environment doesn't play a big role. The species are sort of equal in their capacities. So their distribution is an outcome of processes like speciation, migration, extinction, and so on. So this is what you will predict according to the neutral theory for, let's say, this is for all Antarctic lakes, this is for the Scandinavian lakes, This is when you take the surface only of the lakes, and then when you take the deep waters of the lake. And what we see is that in general, just to summarize this, in general, the fit is quite poor to the neutral theory. And it's hypothesized that if you don't have a good fit to a neutral theory, you need to start thinking about environment having a role. When we started to do some analysis on community composition, we saw that communities that were present in environments with similar salinity tended to have a similar community composition. So this is a UPGMA clustering. What you have here are not species or anything like that. These are communities or samples, let's call it. Here you have Bright-Curtis beta diversity estimator. Here you have a phylogenetic-based unifrac distance. And in most cases, you see that, well, things that come from fresh water, which are in blue, they tend to have similar communities, so they were clustering together. Then brackish samples, they cluster in two groups, let's say, and these were from different lakes. Hypersaline samples from different lakes were clustering. So it seems that salinity is selecting a lot who can live in special places. And what we've done here is we wanted to look at the effects of salinity and also space, geographic distance, in community composition. So here we have Bright-Curtis distance, and here we have the Euclidean distances for salinity, and the distance in kilometers for geographic distance. So what we saw is that salinity has a big correlation with community composition. So if we take all samples, we see that this is a mantle test with a mantle test, and we get like a 0.69 correlation between the salinity you have and the community differentiation you have. Things are not so clear with distance. You have a low correlation between, well, community composition and geographic distance. But then again, these lakes are very stratified, so they are not mixing like many other lakes, which, you know, they mix the whole community over a year. Some lower waters communities might have complete different communities as upper level waters. So we divided the data set in upper surface waters, which can be more influenced by dispersal, and lower level waters, which can be more affected by the environment. So that's what more or less what we saw. I mean, salinity always have a very big role, but if you look at surface waters, you see that salinity has like a 0.5 correlation in the mantle test. And when you move to the deep waters, the correlation increases. And for geographic distance, you see they have a bigger correlation for surface waters and a lower correlation for deeper waters. So you will say, in a conclusion, that salinity always has a quite important role, but then for the surface waters, dispersal might also have a role. Now I'm going to tell you this about the rare biosphere that I mentioned before. The rare biosphere is this concept. Well, if you do this rank abundance curves, this is from a quite famous figure from one guy at our institute, Carles Pedros. You can observe that a few species, they are the most abundant in the community, and then you have this very long tail of rare things which are present in most communities. And this is quite striking, and that's what you see in your high-throughput sequencing data sets. That you get, let's say that 20 species or OTUs which dominate the community and which make up most of the reads, and then you have thousands of OTUs which are really rare. And this is the rare biosphere, and they are poorly known, these microbes. We don't know what they are really doing. There are some hypotheses that they can act as a buffer of the ecosystem. They might be a reservoir of diversity. So I think this is a quite important question now in microbiology. What's the role of the rare biosphere? And now the interesting thing is that all these methods of high throughput, now they allow us to have a look to these rare microbes. So what we've done in the Antarctic lake is that we divided our OTU, or our species table or OTU table into abundant species and rare species. And we've done some analysis, and what we see is that the same patterns that we see with the abundant organisms are present in the rare organisms. So we could say that, at least for this, I mean, the community structuring between these pools has been pretty similar, or like the factors that have been structuring these communities have been acting in both of them. So the rare biosphere, at least in this case, it doesn't seem to be just a random accumulation of resistant or sleeping bacteria which accumulate here and there. They seem to be affected by the environment. Then when we look at these specialists, generalists, habitat specialists and habitat generalists, we use niche breadth, which is defined in general ecology. There's a formal definition of this estimator. There's an equation for this and everything. So we use this estimator to define generalist and specialist. Usually if you have a low B, you tend to be a specialist, and if the B is higher, you tend to be a generalist. So we saw that there are a few habitat generalists, which are here, and many specialists. So this agrees with other observations that come from general ecology, what you see in several communities. And we also saw that generalists and specialists seem to be distributed along abundant and rare taxa. And another thing we've done is, we've been a blast of all our reads against NCBI, and we wanted to see if we were recovering more novel diversity from Antarctica than from our data set in Sweden. That's just to have something to compare with. So this is one of the figures I showed before is percentage of identity of the closest blast hit, and this is the proportion of ODUs. And what we saw is that, there are a lot of things in the, let's call it the known diversity zone. Then this is related to known things. And there's then this tail that could be novel diversity that's found in Antarctica. We've been doing some work with the OTUs present here, and it seems that in quite a few cases there might be some diversity which is quite novel and from Antarctica. Conclusions for this Antarctic lakes part. So salinity had an important role in shaping communities. Most communities differ from the ancestral marine communities. Abundant and rare taxa seem to have been affected by the same structuring forces. We saw many habitat specialists and a few generalists, which is, agrees with patterns seen in general ecology. And some novel diversity seems to be in lakes. Now, let's wrap up a bit the whole thing, because it's been like an hour and 15 minutes now. And if we go back to the general view, high throughput sequencing and the whole thing, there are many exciting new opportunities in many fields. So this is like an open door, and there are many things that can be done. Probably we need to use our imagination and see what questions we can answer with this new technology. There seems to be a co-evolution of sequencing technology and bioinformatics. This is really interesting. New tools are needed to use all this new data. Now there's a closer integration between biology, computer science, and statistics. So now biologists, we need a much stronger computer background, and probably a lot more on statistics. And in what I see, I think is in the next years, there will be an overwhelming production of data, which will be pretty huge. So we'll be in a situation like this. We have like a wave, a huge tsunami of data, And we'll be like, or hopefully we'll be like this guy, a surfer, and not just sinking in the water. So it's going to be a lot, and it's going to be a challenge. But, you know, things can be done. We probably need to endure this shock. And yeah, I mean, there's a great future ahead. So I want to acknowledge quite a few people that gave money for us for all these projects. And I want to thank all of you for this long presentation. Thanks.